Welcome to hybrid seminar for spring 2023. Nice to see everybody in person. Nice to see, actually the ratio of in-person to online is greater than 1, which is really a good sign. Really happy to have everybody here in whatever mode you're in. So today's speaker is Dr. David Ensminger, who is starting his 4th semester at San Jose State. It goes fast, don't blink. A little bit of a bio on David. He did his bachelor's degree in marine biology right over the hill in Santa Cruz. Is that where you got introduced to elephant seals? Okay, good. Then he went to Sonoma State, which doesn't spring to mind when I think marine biology, but he also studied marine biology at Sonoma State. Then he went and did a PhD in the Department of Wildlife and Fisheries Sciences at Penn State in ecological physiology. Postdocs briefly at Western Kentucky and then at UC Berkeley. And then he came and joined us here a little less than 2 years ago. He studies stress physiology in wild invertebrates, which is a great category if you want to study lizards and elephant seals. And I don't know, and I guess it includes lots and lots of things in that range. So today's talk title is Transgenerational Impacts of Stress in a Wild-Caught Lizard. For those of you who are online, if you have a question, go ahead and type it in the chat. If you are asking something that we think David should answer right away, I'll interrupt him. Otherwise, we'll leave the question for the end and we can answer it live. Okay, and those of you in the room know how to raise your hands, so we're all good to go. All right, so David, take it away.
Okay. Well, thank you everyone coming to my talk. I'm excited to tell you about some of the research that I've been working on and we're going to be continuing to work on in my lab. So we're going to start the talk off with a little bit about general stress physiology. How does it work? And in particular about a key hormone, glucocorticoids, which are important metabolic hormones, but when elevated to stress-induced levels, they have a myriad of different impacts on organism. And next we'll talk about how those then impact the next generation, the offspring of the individual who is being stressed. And finally, we'll talk about some of the future studies that my lab's currently working on. Now, animals experience a myriad of different stressors throughout their life. Things like lack of resources or challenging climate conditions, but also ecological interactions such as predation and conspecific interactions. But in addition to these stressors that these animals are experiencing from their wildlife environment, humans are also adding more stress to these environments. Things such as strip mining and clear-cut foresting, or even more pertinent to current news, climate change or invasive species. So these additional stressors are compounding onto what animals normally experience, which really makes it important for us to understand what are the impacts of these stressors. But all of those different stressors are integrated into a physiological response through the HPA axis, or the hypothalamus pituitary adrenal axis. When stimulated, the hypothalamus releases corticotropic releasing hormone, or CRH, which stimulates the anterior pituitary to release ACTH, adrenocorticotropic hormone, which then circulates through the body, interacting with the adrenal glands to release many different hormones and metabolites, but one of the key ones being glucocorticoids, also known as CORT. And CORT at its baseline levels, as I said, is a metabolic hormone. It increases lipid degradation and protein degradation to release energetic molecules. But it's also important for things like biorhythms. Your sleep schedule is dictated by this hormone. But when it's elevated from baseline levels to stress-induced levels, we start seeing some different impacts. We're going to have massive changes to behavior, causing increases in anxiety-like behavior, such as hiding in a corner or hiding underneath something, increased stigmataxis, the desire to be to a wall. And people have also found that when CORT is elevated, you are more likely to respond to a stressor as well. You respond quicker and stronger, as shown here, where a factor showed in lizards, when he added CORT to these lizards, they responded much quicker to a novel object. And all of these behavioral changes and physiological changes are made to promote immediate survival. So instead of giving energy to your reproductive system or immune system, it's trying to get you away from the thing that's going to end your life right then. Because it doesn't matter if you can reproduce if you're not alive. But it also does have massive impacts on reproduction. So here with adult male northern elephant seals, we showed that during the early breeding season, when it's incredibly important for these males to have high levels of testosterone to promote spermatogenesis and allow them to have antagonistic interactions with other males to get territory for breeding. After 2 hours of stress, testosterone has decreased by about 15%. But after 48 hours of stress during early breeding, testosterone is less than half of what it normally is. And this is the time when they really need that testosterone to have proper reproduction. But it goes beyond just reproductive hormones and actually influences reproductive success. With Kirsten McLeod, we showed that for lizards who had CORT administered to them, the hatching success of their eggs was decreased as well. So it's affecting the hormones coursing through the individual, such as the reproductive hormones, but also just the ability to properly reproduce. But these are all activational impacts of stress, how CORT impacts the individual. If this happens during gestation or development, we can also start to see some organizational impacts of stress, where they change how an organism is developing and organizing their physiological systems. So if you have a mother who's pregnant, she has some baseline level of glucocorticoids circulating through her for the metabolic impacts. And these glucocorticoids are lipophilic. They can pass through many different membranes, and they'll pass through the placental barrier to interact with the offspring. But in placental mammals, there's an enzyme in this barrier called 11-beta hydroxysteroid dehydrogenase, which inactivates some of the CORT so that the offspring only experiences a subset of a mother's glucocorticoid concentration. However, if a mother experiences a stressor, upregulates the amount of glucocorticoids going through her body, this can start to overwhelm that 11-beta hydroxysteroid dehydrogenase barrier, and more of the biologically active form of CORT gets introduced to the offspring. Interestingly, though, in organisms that reproduce without a placenta, such as egg-laying lizards, there isn't this 11-beta HSD enzyme. They just experience the full front of a mother's CORT response. And for a long period of time, people looked at the effects of maternal CORT on the offspring and thought this was negative. You're making a worse baby. And they thought that because it increased fearfulness, it made animals more skittish, it increased anxiety levels, forced them to hide more. So we have massive changes in behavior, but it also led to the production of smaller offspring and offspring that grew slower. So you have these anxious babies who are smaller and growing slower, and people thought this is negative. You're having worse babies as a side effect of the mother surviving that stress encounter. So stress helps the mom survive, consequences get passed to the offspring. But is it really decreasing the fitness? Is it really making bad offspring? And to understand that, we can't look at these changes in a laboratory scenario. We have to think about where these changes would be occurring in ecology. So a mother isn't just going to randomly be stressed. Something's going to stress them, such as hybridation risk, that elevate maternal glucocorticoid concentrations, which then alters the offspring phenotype, changing morphology, behavior, and physiology. And if we want to understand if these are beneficial or detrimental, we have to look at how that allows the organism to live in the environment that it's going to be in now, which typically is the same environment that the mother experienced. So people started questioning whether or not maternal stress was bad, and they started doing some experiments. And Love and Williams found in starlings that offspring from mothers who had high CORT concentrations had a smaller CORT response to a novel stressor. They're more resilient. They don't mount as large of a stress response. And Storm et al. Found in crickets that offspring crickets from a stressed mother had increased anti-predator behavior as well. And the way they did this was they increased the time spent immobile when a predation event occurred, because how they hide from predators is in plain sight by blending into their background. So if they're not moving, it helps protect you. So this makes it sound like to me that maybe maternal court is adapting an individual to a stressful environment, which if you're working in the lab, which is a control environment, that would look maladaptive. I was really curious. I wanted to dig deeper into how does maternal court change the phenotype of the offspring. So working with Tracy and Kilden in Penn State, we looked at this in eastern fence lizards and wanted to examine how maternal court changes hormone, nutrient, and calorie allocation to the egg, as well as the morphology, behavior, and physiology of the offspring coming from those eggs. So we use the eastern fence lizard because they are easy to catch. We know a lot about the ecology, so we could put all of these changes into an ecological framework. And they have discrete eggs that we can measure hormones, nutrients, and calories from easily. And we know a lot about what their day-to-day life is, what type of stressors they experience. They have a lot of predation events with visually hunting predators such as birds and snakes. But as of 80 years ago, they also have to contend with a new predator, the invasive fire ant, which was introduced to Port Mobile, Alabama about 80 years ago. And they're slowly adapting to, but it's still, in terms of evolution and adaptation, a novel predator. But on top of those stressors, they're ectotherms, so they have to deal with thermal stress and how they utilize the environment, and competition as well. They need to compete for basking sites and food storages, as well as areas to reproduce. We get a little bit of all these different stressors. And to do this work, we went out to the field with a fishing pole attached with a little noose on the end, and we go capture the lizard and then bring it back to the lab. And once in the lab, we would at night administer our court treatment. And this was done non-invasively through a transdermal application of an oil. So we could do this while they're sleeping without disturbing them. And the nice thing, this elevation in court was to an ecologically relevant level as well. So we're getting away from the issues with implants, the elastic implants or injection, where everything was ecologically relevant and we're not disturbing their sleep.
Can I ask a naive question?
Yeah.
Doesn't catching them on the end of a fishing pole increase their court level?
It does, which is why that would be a minor stressor for all of them. And they normally experience a couple of those a day. That one stressor, bring them back to a control environment. Now, the control ones are getting no stress, and the other ones are getting one stressor a day.
Okay, thanks.
Yep, good question. So after we did these treatments from capture to oviposition, we started doing some measurements. And the first thing we wanted to look at was, did our hormone actually get into the egg? And we found it did. This is wonderful news. Our treatment's working. It's not only getting the hormone into the mother, but it's also going into the egg as well. So from there, we looked at nutrient compositions, and we found a massive decrease in the amount of protein being allocated to these eggs, and a trend for an increase in the amount of lipids being allocated. You might be asking why carbohydrates aren't here. It's because it's been assumed and slightly shown that reptiles don't really have many carbohydrates in their eggs. So due to the small sample size, we focus more on these. And when we combine that together, we see decrease in protein, a little bit of increase in lipids. There actually wasn't a difference in calorie content. And while that makes a little bit of sense, because lipids are so much more calorically rich than proteins, the math isn't quite there, which makes me think we might have to reassess whether or not there are carbohydrates in these eggs. There is no albumin, but maybe carbohydrates are mixed in the yolk. But we saw this decrease in protein, and we're thinking, hey, maybe this is why offspring from stressed mothers are born smaller. They have less of the building block, less protein. And that would have been a really nice story if that actually came true. But with fence lizards, offspring from stressed mothers were born larger. They had longer bodies, longer tails, and their mass was a little bit bigger as well. So this is completely counter to many different animals. And things like rodents or hares or even humans, stressed mothers generally have smaller offspring. So this is a little bit confusing for a while, and I went through the literature trying to see if there's any evidence of other organisms having a similar response. And Dancer et al. Found in red squirrel, they had a similar response. Stressed mothers would give birth to larger offspring. I'm thinking, all right, what is similar between these organisms? One's a reptile, ectotherm, one's an endotherm. But both of them have a lot to do with competition, conspecifics with each other. Red squirrels, their biggest stressor is overcrowding. They have to compete for space, they have to compete for food, for reproduction, for where to hibernate. Everything is competition for them. And so if a mother makes a smaller offspring in a overcrowded area, that offspring is not going to do well. It's going to be outcompeted by the other mothers. So maybe evolutionists selected for larger offsprings, better competitors, and that way the mother still has fitness. And that might be what we're seeing here as well with the fence lizards. One of their chief stressors is competition with each other, so potentially this could be beneficial. But if we go one step beyond that, we might start to see something interesting with their new predator as well, the invasive fire ant. The fire ants will bite and envenomate the fence lizard with their venom, paralyzing them, and then at that point they consume them. But if you're larger and grow faster, you're now bigger. So by simple body dilution, it's going to take more venom to incapacitate you as well. So we might see a further selection for this increase in body size due to this invasive fire ant if there's a correlation here. But from there, we went and we looked at embryonic heart rate. What was the heart rate of the embryos inside the eggs? So working with Dustin and Hannah, we found eggs from court-stressed mothers had an increased heart rate compared to eggs from control mothers. And at this point, I thought, not only awesome, we have high metabolic rates, all those fun things, it finally made a connection to me. Because do it all has shown in many reptiles that if you increase the heart rate of them, you increase developmental rate. It's a biological clock. After a certain amount of heartbeats, they hatch. So if we have an increased developmental rate, and incubation times were a little bit shorter as well, they could be using more of this protein instead for maintenance, they're using it for actual development. So that might be what's allowing these organisms to have less proteins but be bigger, is because they're speeding up development, utilizing more of that protein to just grow bigger faster. And that would be beneficial in a stressful environment. You're out of the vulnerable egg stage quicker. But as many things in biology occur, if there's a benefit to something, there's probably a cost as well. And I believe one of those costs might be developmental errors. As you increase developmental rate, hypothetically, you get more errors as well. And we're still collecting data on this, but the preliminary data is showing that. We've seen lizards born without any tails. We've found lizards born with a question mark tail. We've seen them born without a right ocular cavity, straight bone with flesh over top of it. And finally, there's a lizard that we nicknamed Flippy, because whenever Flippy was stressed out or scared, instead of running around or hiding somewhere, he would sit there and do backflips. So there's probably something neurologically interesting with that lizard. But this is something, as we keep doing this work, we might find more and more get to a statistical significance for this result. Yeah.
Do you see more of them die? Like higher lethality in that group too?
Not yet. So there is higher hatching success and control, but once they're hatched, we haven't found any difference in survival. In the lab, at least. So, as we talked about behavior, next thing we looked at was with anti-predator behavior. So working with Michaela and Nud, we looked at time spent underneath a perch hiding, and time spent, or how long it took them to respond to a tactile stimulation. And we found increased amount of time spent hiding underneath the perch, which would make sense if your chief predator is visually hunting birds. The more time you spend out of the open, the less vulnerable you are to those predators. And a similar thing with tactile response. One of the ways that these animals also avoid predators is through crypsis. And as you can see, that lizard's pretty well blended into the environment. And if they stay still, they will continue blending into that environment. But as soon as they move, they're going to be incredibly visible to any predators that are there. So by decreasing their likelihood to move when something touches them, that could increase that anti-predator behavior. If something like a common ant climbs on their foot, they're less likely to freak out and move. But they don't have to just deal with common ants at this point. This could be an evolutionary trap where staying still might be beneficial against their visually hunting predators. This could be a maladaptive trait against their new predator, the invasive firehead. So this will be interesting to see if this actually persists as their interaction stays together. But not everything about stress should be focused on just anti-predator behavior. Risk is a huge important part for how organisms behave. There's a whole field called the landscape of fear that looks at that, but it's not everything. When we think about behavior, we also have to think about habitat selection. Where do these organisms live? What do they do there? And two of the key drivers of that are temperature and humidity. They dictate a lot of where any vertebrate is, especially for an ectotherm. So my master's student, Amber Singh, is working on this project right now, where she wants to look at how maternal and acute stress can alter habitat selection. So we're going to capture our gravid females. Some of them will be controlled, some will be court. We'll look at how they utilize different areas. But then we're going to take two offspring from each of these mothers. One of them will remain a control, and the other one will get the acute stressor that the mom had been getting as well. And by having this kind of two-factorial design, we can make a lot of comparisons together. We can look at how maybe just age affects habitat selection, or individual stress, or how age and individual stress interact, or the interaction of maternal stress and individual stress. And to look at these and examine this, Amber's been building this behavioral enclosure that will allow us to manipulate temperature and humidity while getting access to both of those to the lizards. So we have our heat lamps on the y-axis, high temperatures at top, low temperatures at the bottom. And across the x-axis, she's found a substrate that will hold humidity constant that we can vary between low, medium, and high humidity levels. And that's what we can see. Does the organism like basking closer or hiding further away across this gradient? And her project is really going to help us understand the connections between developmental physiology and how these organisms behave. We've always known that an organism's own physiology affects their behavior. That makes sense. But now we're going to show even more so maternal physiology while you're pregnant can affect the offspring as well. It's going to be really the first examination of how maternal physiology can interact with an individual's experience, the offspring's experience, to alter their behavior in wild reptiles. But when we look at behavior and we try to extrapolate beyond right at that moment, it gets a little bit extrapolating. We're drawing hypotheses there. And the key question is always, how is this affecting fitness? But fitness is really hard to study in wild, especially organisms that live multiple years, because to study fitness, you have to look at the contribution of an individual to the grandchildren's generation and compare that genetic contribution to the entire population. As you can imagine, that's going to be complicated. So there's a few different ways that we can make little proxies, though. And one of them is looking at survival. If you're not alive, you can't reproduce. So hypothetically, if we'd say maybe control offspring live longer than control, or sorry, court offspring live longer than control offspring, maybe there's an increase in fitness there. So we tried doing that type of experiment. And in South Alabama, we did an outdoor enclosure experiment for about 10 days. Granted, 10 days doesn't seem long, but in an area that is frequented by many different storms, having aluminum flashing in closing areas isn't really conducive to massive torrential downpour. But what we do with this is we made a two-factorial design where we've had offspring from control mothers and offspring from court-treated mothers. But then we also manipulated the environment. We had areas that we removed the fire ants from. And the way we did that was with a steel rod and boiling hot water. Pound the steel rod into the fire ant mound, pour the boiling hot water in, we eradicated that fire ant mound without poisoning anything. And then we have the enclosures where we left the fire ant mounds in there. And there was over 20 fire ant mounds per enclosure here. So natural levels that are pretty high still. And what this full factorial design really allowed us to do was assess the hypothesis known as the mismatch hypothesis. And what this states is, if, let's say you have a high predation risk environment, that's going to elevate maternal glucocorticoids, alter her offspring as we just talked about. And if that offspring goes into a high risk environment, that should be beneficial, adaptive. There should be increased fitness there. But if that same offspring goes into a control environment, such as a laboratory, there should be consequences that should reduce the fitness of that organism. And this is really allowing us to make some predictions on survival. Control offspring in a non-stressful environment should do the best. There's no changes, there's no additional stressors to them. They should survive pretty well. Offspring from court-treated mothers in a control environment should do next to the best. There might have some fitness consequences of the physiological changes due to the maternal court, but there's not high predation risks here. A little bit worse than that will be the court-treated offspring in a stressful environment. They have high predation risks, so it's going to be a little bit of a decrease, but they're prepared for that environment, so they should do all right there. Compared to our control offspring going into the mismatched high-risk environment, who should be completely naive and at that point at high risk. And this also throws in some economic theories, such as hedging and error management theory, which would state that at this point, mothers should start making control offspring because at least they're going to be doing well, as opposed to potentially all of them dying in a high-risk environment. And when we look at the actual data we got from this, working with Cameron Venable, we showed that in a low-risk environment, offspring from control mothers had high survival. All right, that makes sense. Perfect. And offspring from control or from court-treated mothers had decreased survival, but they're still doing pretty good. So first two steps, check. But when we look at the high-risk environment, we see something kind of interesting. We see offspring from court-treated mothers have the same survival. They are unfazed at 10 days due to the impact of fire ants and high risk. And it's not just that we maybe aren't seeing any risk impact, because our control offspring have decreased in survival. So I think if we were able to increase the time that we were able to do this enclosure to maybe 5-10 weeks, we might start to see a teasing apart of these two, in which case it would perfectly map onto the maternal mismatch hypothesis. And the reason I give that suggestion on why we might start to see it if we lengthen this is because when Kilda and Friedenfeld found that when exposed to fire ants, the survival impact is a little bit delayed. Here on the top line, we have control individuals just given regular ants, and their survival is pretty good out to 16 weeks. But when we look at lizards who have been either stung or fed fire ants, survival decreases on average about 3 weeks. And with this enclosure experiment, we were looking just at these first 10 days. And we're already starting to see some separation. So I really do think if we increase this, we'll see more and more of this. But like I said, Alabama likes flooding, so it's a little bit harder. So survival is one aspect, and I just said it's hard to do long-term enclosure experiments. I was trying to figure out, is there a way that we can estimate long-term health and survivability? And some people have suggested that telomeres can serve as a proxy for long-term health. Telomeres are these protective end caps on the ends of chromosomes that promote chromosomal stability and proper replication. And as an organism ages and the cells go through replication, the last little bit of these telomeres gets shorter and shorter and shorter due to what's known as the end replication problem, which states simply that the last little bit of chromosomes cannot be perfectly replicated, so it decreases in length. Interestingly, that decrease in length has been correlated with increased disease risk, decreased health, and decreased survival across many different taxa: mammals, reptiles, fish, birds. So if you are stressed, it's increasing your metabolism, as we talked about, which increases your cellular turnover. So you get this end replication problem more and more. Additionally, though, stress can also increase oxidative damage, which can shorten telomeres prematurely as well. We have two routes that telomeres could potentially be shortened due to stress. So working with Sue Siegel, we found that in sons from CORT-treated mothers, they had shorter telomeres than sons from control-treated mothers. And this is seen in contrast to daughters who were not impacted, whether they came from control or CORT-treated mothers. I thought this was interesting. We're seeing a sex-specific impact on telomere lengths due to maternal stress. And that could potentially have a couple different drivers causing that impact. Many different animals and people have shown that there's differential investment sometimes in the offspring. Mother birds will sometimes prioritize giving food to the female offspring over the male offspring in order to maximize their fitness gains. And this follows the ecological theory on Trivers-Willett hypothesis, saying if there's a fitness benefit from one individual to another, the mother should prioritize that one. And with ecology, the females are always going to reproduce. Sons are a risky investment, so they should prioritize the daughters. And maybe that's what's happening. There is differential investment of things like antioxidants, which could protect the offspring that we've seen. Alternatively, though, it could have less to do with mother investment and more to do with just the sex of the offspring. Because estrogens, one of the key female reproductive hormones, increases not only antioxidants, which can protect you from oxidative damage, but it also increases telomerase activity, which can slow down the degradation of telomeres. Conversely, testosterone does the opposite. Testosterone decreases antioxidant capacity as well as decreases telomerase activity. So maybe it's more to do with just the individual. But either way, shorter telomeres have been suggested to lead to decreases in health as well as survival, suggesting maybe there is a fitness cost to the sons of maternal stress, but not the daughters. So I hope today that I've been able to show you that maternal court might be preparing the offspring for a stressful environment, as we saw with things like the behavioral changes and the morphological changes, but it might not necessarily be adapted to a novel stressful environment, such as an invasive species. And many different stressors can integrate to elevate glucocorticoids. Things like predation, environmental, or anthropogenic activities can all be integrated into this physiological response. And so I think really CORT can have massive conservation implications because, as we talked about, it can influence reproduction. So we have decrease in hatching success of CORT-treated mothers' eggs compared to control-treated mothers. But beyond that, it can impact survival as well. Bless you. Here we have survival of control mothers from 2015 and 2016. And when we look at the survival of cord-treated mothers in 2015, it's about the same. It's relatively close. But when we look at 2016, we see survival massively dropped here. And our methods were the same. Everything in the lab was the same. The weights that we caught them at were relatively similar. So we're wondering what caused this almost 50% decrease in survival of these individuals. And when we look back at the data, we found the winter between 2015 and 2016 was incredibly warm in the southeast that year. And reptiles, when it's colder, they go into a state of torpor. They lower their metabolic rates with the lowering temperature, so they don't have to worry about getting as much food because there's less insects out during the winter. But if it was warmer during that winter, they wouldn't be able to lower the metabolic rates as low. They go through more of their stored energy, their fat supply. They would come out in worse body condition, which could potentially predispose them to adverse stress reactions. So here we see kind of the compounding of different stressors across time as well. And finally, we've also shown that fawns that had higher court in general had lower survival in the same environments as well. So it's really important to understand the survival and how organisms are interacting with their environment. So earlier we talked about some of the work that my current master's student Amber is working on. And we're also going to start exploring more of the molecular and cellular side of the maternal response to stress. In particular, I want to examine more on the oxidative stress and the impact to immune function. So, as we talked about, glucocorticoids can change metabolism and liberating energetic molecules. And both these metabolic hormones, as well as glucocorticoids, can change cellular function. In particular, they can increase reactive oxygen species. And these reactive oxygen species at baseline levels are important signaling molecules for a variety of different cellular functions. And typically, they're counterbalanced by antioxidants. So you have the signal, and before that reactive oxygen species can do any damage, it's nullified by antioxidants and protected. The problem, though, is that stress increases reactive oxygen species generation, which can throw off this balance. So now you have more reactive oxygen species than you have antioxidant capacity, which can lead to oxidative damage at a cellular level, disrupting lipids, proteins, and carbohydrates. So I was really interested in this. I want to see if stress does that. What about maternal stress? So what I wanted to do was a live cell imaging to examine the oxidative response to our stress hormone, and also a general oxidant, tert-butyl hydrogen peroxide. CORT would allow us to see maybe there's a hormonal difference in how the offspring would respond to oxidative insults. And this would allow us to see maybe there's just a baseline different physiology of the offspring altogether. But to do live cell imaging, you have to first get live cells. So people have been isolating cells from many different organisms. Some people are working on things like bats and whales. Not many people are doing this with reptiles, though. And there's a bunch of different challenges. But working with Jose Pablo Vasquez Medina and Gabriela Arango from UC Berkeley, we've been able to isolate these fibroblasts here at San Jose State University and stain them for two different markers of fibroblasts. PDGF here in red and vimentin here in green. And blue is our little nucleus. So we can isolate our cells. We've shown that we have fibroblasts, which are a key structural cell, ubiquitous throughout the body. The next step is to do the live cell imaging, which we're working on now.